Good afternoon. First of all, thank you for the invitation. It's a really honor for me. Today, I would like to talk about global issue, antimicrobial resistance spread in environment, especially in extreme environment, focusing mainly on the results of my work of the work nominated for the Frontiers Planet Prize. Therefore, the title of my speech is Arctic Plasmidome Analysis Reveals Distinct Relationships among Associated Antimicrobial Resistance Genes and Virulence Genes along anthropogenic gradients. And the emergence and spread of antimicrobial resistance is among the greatest threat to global health, according to World Health Organization. Currently, drug-resistant disease causes over 1 million deaths globally per year, and it is estimated that this number can increase to 10 million deaths globally per year by 2050 if no action is taken. In this figure, you can see that this number is comparable to the global cancer death toll in 2020. And here, in this illustration, you can see that the prediction of global death that's from antimicrobial resistance in 2050 looks like if no action is taken. So, as you can see, this is a very dangerous phenomenon on a global scale. And generally, the overuse and misuse of antibiotics not only for human therapy, but also for livestock breeding and agriculture, are likely major contributors to the emergence and spread of antimicrobial resistance in environment. I mean, antibiotic-resistant bacteria and antibiotic-resistance genes in environment. And this can happen in environmental. So this can happen in environmental media, in water and soil, and from sources such as wastewater, from wastewater treatment plant, which is a hotspot for antimicrobial resistance spread, where there is a high abundance of bacteria for various sources, nutrients, heavy metals, detergents, and antibiotics in subinhibitory concentrations. And the presence of antibiotics in concentrations lower than the concentrations inhibiting bacterial growth contributes not only to the selection of resistant strains, but also to the increase of the frequency of horizontal gene transfer between strains of the same or even different species. And consequently, this leads to the appearance of resistant bacteria also in effluent, which are discharged into surface waters. And these bacteria can belong to critical pathogens with the most dangerous mechanisms of resistance that pose the greatest threats to public health, according to World Health Organization. With increasing human activity and climate change, human activity, I mean overuse and misuse of antibiotics and release other pollutants, and climate change changes in the cryosphere accelerating the spread of antimicrobial resistance through aquatic environments, crossing planetary boundaries and impacting novel entities. And despite the increasing number of publications on the mechanisms of resistance, antibiotic resistance spread, little is still known about this phenomenon in polar and rapidly changing regions. Literature data suggests report that approximately 10 to the 21st power cells are released from melting glaciers worldwide. And the state of knowledge about the released antibiotic resistance determinants is completely unknown. So the Arctic environments provides the opportunity to observe the combination effects of increasing human activity and climate change on antibiotic resistance spread in environment. And a very interesting structures in the context of antibiotic resistance spread in environments are integrons. Contemporary studies show that integrons are mainly responsible for the emergence and spread of multi-drug resistance among bacteria. Integrons are DNA fragments that are capable of capturing genes cassettes encoding for antibiotic resistance and resistance to disinfectants and heavy metals. So integron system can create novel combination of genes and facilitate the spread of antibiotic resistance and multi-drug resistance among bacteria, as I said before, because it is essential that integrons are embedded in mobile genetic elements and such as transposons and plasmids, and can be transferred between bacteria and microbial population by horizontal gene transfer. I have to mention here that integrons are evolutionary old structures and occur in the genomes of bacteria for various sources. And we know 5 classes of integrons associated with antimicrobial resistance genes, but Class 1 integrons are particularly important in the spread of multi-drug resistance, and due to widespread presence in the genomes of commensal bacteria for human and animals. They high frequency in wastewater and highly conserved DNA sequence. Class 1 integrons are recognized as a great environmental marker of pollution associated with human activity. And taking into account the main aim of our study was the directly evaluation of impact that water flowing from melting glaciers and wastewater discharge directly to the fjord into Arctic region exert on increasing the genes pool of the global aquatic resistome. More specifically, the research aimed to explore antibiotic resistance spread across human-affected and pristine environments by comparing various aspects of the plasmidome. And here I have to mention what's exactly it's a resistome. Resistome is the collection of all antibiotic resistance genes in environment, including those associated with pathogenic bacteria, non-pathogenic antibiotic-producing bacteria, and other antibiotic resistance genes. And most antibiotic resistance genes are located on mobile genetic elements, such as plasmids, transposons, and integrons, which constitute the mobile part of resistome, called the mobilome. And the plasmid portion of the mobilome is called plasmidome. And the plasmidome, so the plasmidome is a mobile part of resistome and is especially significant in bacterial evolution, especially with respect to horizontal gene transfer of antibiotic resistance genes. Anyway, we determined the abundance and diversity of antibiotic resistance genes and other stress response genes, and also integrons and virulence genes in Arctic environments. Samples were collected from Spitsbergen and the largest island in Svalbard archipelago during 3 summer seasons. And polar region, free of direct and permanent delivery of contaminations, are target places for tracking human impact in natural ecosystems. Owing to this simplicity and the different human pressure, I mean glaciers, city, wastewater, fjord, we choose this area for studies on the effects of the melting glaciers and human activity on the resistance of freshwater and marine environments. To investigate this impact on the resistance, sampling was conducted in contrasting system along 2 gradients of anthropogenic influence. First, the Longyearbyen city gradient. Longyearbyen is the capital of Svalbard. The gradient, the transect started from glaciers, river water flowing from melting glaciers, and untreated wastewater directly to the fjord, and fjord before and after discharge of untreated wastewater. And the second gradient was wastewater treatment plant outflow in Nalesund. Nalesund is a small city where scientists and research stations are located, and of course wastewater treatment plant, because this is a difference between these 2 cities, between Nalesund and Longyearbyen. Where isn't a wastewater treatment plant. So the sampling was collected from raw sewage, effluent, and water from fjord, which is the effluent receiver. As a reference sample, so we collected ice cores from Northern Skjoldbren, which is a non-touristic and significantly larger glacier than the 2 glaciers from first gradients, city gradient. And in my research, I use various approaches based on culture-based methods and metagenomic approach, in which I analyze total environmental DNA. In this particular study, I focused only on the plasmidome, so this is only part of my research. And what was the research plan? Firstly, I wanted to isolate environmental plasmidome, and it was really challenging. It was really difficult because direct isolation from environmental samples gives us very low concentrations of plasmidome DNA. So I spent a lot of time trying to standardize these methods, and finally, we used an enrichment step in the medium before plasmid isolation, which gave us the best results of concentrations of plasmidome DNA. We then isolate plasmidome DNA and sequence them. And what we found not only antibiotic resistance genes, but also other stress response genes, including metal and biocide resistance genes, and virulence genes, and of course integrons in plasmidome DNA. Total 67 are antibiotic resistance genes were identified in the plasmidome across all sampling sites, with most conferring multi-drug resistance by efflux pump and resistance to beta-lactams and other clinically important antibiotics. Here, I have to mention that we found that the most common antibiotic resistance genes were the BLA1 and BLA2 genes, which were detected in samples regardless of the levels of the anthropogenic input. What's most important is that the BLA2 gene is a metallo-beta-lactamases that catalyze the hydrolysis of a wide range of beta-lactam drugs, including carbapenems, which are considered the antibiotics the last resort in the treatment of the serious infections, of the most serious infections caused by gram-negative bacteria. So it's a very dangerous resistance mechanisms in almost all sampling sites. Next, metal resistance genes. A total of 41 metal resistance genes were identified in the plasmidome, conferring resistance to 10 metals, with the dominant resistance to mercury. A total 3 biocide resistance genes encoding resistance to quaternary ammonium compounds and multi-drug efflux pump. And total 26 virulence genes were identified across the plasmidome, and the production of exotoxins, including enterotoxins, important for bacterial pathogenesis, was the dominant virulence mechanisms identified in this data. And we found that there was a significant increase in the relative abundance of both virulence genes and stress response genes, including antibiotic resistance genes in water, in fjord water, of Adventfjorden, after discharge of wastewater, compared to water and sediment of fjord seashore. So this suggests that wastewater discharge significantly increases the gene pool in the aquatic resistance, contributes to the antimicrobial resistance spread in the environment. Here you can see the prevalence of antibiotic resistance genes and metal resistance genes classes detected in the plasmidome from environments with different anthropogenic pressures. And we found that the diversity of antimicrobial resistance and the number of classes of antibiotics to which the antibiotic resistance genes encoded resistance increased with increasing anthropogenic input, and water from the river flowing from melting glaciers, as well as in raw sewage environments, contained the greatest diversity of metal resistance genes, which indicate that the melting glaciers could be reservoirs and potential sources of antimicrobial resistance determinants entering aquatic environments. And what the relationships between the glaciers look like we found that the frequency of detections of virulence genes decrease along the gradient from the glacial environments through the river flowing from melting glaciers to the fjords, and significantly increase with the level of anthropogenic input after discharge of wastewater. This indicates that also a potential role of glaciers in releasing virulence genes. Literature data suggests that there is on the glaciers that is strong pressure from viruses on heterotrophic bacteria on glaciers, which may affect the pathogenicity of bacterial communities by the transfer of viruses encoded virulence genes. Maybe that's why. And additionally, bacteria released from melting glaciers may have virulence genes homologies and are unlikely to be opportunistic human pathogens. And these genes may be mentioned for benefit or for co-occurrence with other genes conferring benefits for bacteria survive outside for the host environment. We found that the frequency of detections of RGs, antibiotic resistance genes, increased in river water flowing from melting glaciers and in the effluent from wastewater treatment plant in Nea-Ålesund and the fjord after wastewater discharge, both Longyearbyen and Nea-Ålesund, whilst the detection of the integrons in fjord was most frequent in the water receiving effluent from wastewater treatment plant in Nea-Ålesund. And the highest frequency of detections of metal resistance genes was found in raw sewage from a wastewater treatment plant in Nea-Ålesund. And another interesting finding was that the relative abundance of antibiotic resistance genes was strongly correlated with metal resistance genes, but not with integrons. And this is a really interesting finding. While virulence genes correlated with biocide resistance genes. It is worth mentioning that in our study we found that the integrons located in the Arctic plasmidome mainly contain adaptative genes and genes for hypothetical proteins with unknown functions. Because resistance genes we found were only identified in wastewater, which points to a relationship between antibiotic resistance genes and anthropogenic input. Our results suggest that stress response genes, including antibiotic resistance genes and biocide resistance genes and metal resistance genes, and also virulence genes, are present in the plasmidome, but outside the structure of integrons. We didn't find typical antibiotic resistance genes in structure of integrons, suggestions that they may not play as important role in the spread of antibiotic resistance in the Arctic as previously thought. So it's really interesting finding. And principal component analysis revealed that biocide resistance genes correlated with virulence genes dominated in glaciers, and antibiotic resistance genes, which are correlated with metal resistance genes, dominated in wastewater receiving effluent, receiving water, and water flowing from melting glaciers. Additionally, integrons are dominant in samples from environmental with varying levels of anthropogenic input. And what can we say about these results? First of all, mapping the occurrence of plasmid encoding genes in Arctic ecosystem provide key information about towards understanding the sources and pathways of spread of antibiotic resistance genes and informing efforts to mitigate the spread of antibiotic resistance. And conclusion, first conclusion, plasmid encoding antibiotic resistance is widespread in the Arctic environments where selection pressure for antibiotics is presumed to be low. This resistance may originate from both anthropogenic sources as well as the release of ancient variants of genes from the melting glaciers. The presence of metal resistance genes on plasmid combined with the prevalence of metals in the environments could potentially facilitate the spread of resistance by horizontal gene transfer, because a strong correlation between metal and antibiotic resistance genes confirms their co-occurrence on the same mobile genetic elements and can create the opportunity for co-selective pressure. And in the glacial environment, virulence genes and biocide resistance genes predominant, which suggests that glacier ice may be a repository of pathogenic strains, with associated genes on the plasmidome. And the widespread occurrence of genes determining physiological functions of genes for hypothetical proteins with unknown functions in variable region of class 1 integrons highlights a central role for these genes in adaptation of bacteria to Arctic environments, but also presents a risk of developing new resistance phenotypes. And the absence of antibiotic resistance genes in structure of class 1 integrons in the plasmidome may also indicate unique features of the microbial community in the extreme environments. And so this is a huge threat to human health, animals, and the environment in the future also. And finally, and this is my research team, and without them, this article wouldn't exist. So thank you. And thank you all for attention.